1. Spore Dispersal. The Sporophyte's capsule dries out and bursts, releasing haploid spores into the air. These spores eventually settle on a moist, suitable substratum. 2. Primary Protonema Formation. The spore germinates directly into the primary protonema. This is a creeping green, branched and frequently filamentous stage that spreads across the soil surface. 3. Secondary Protonema Formation. The primary protonema undergoes fragmentation or produces specialized branches to form the secondary protonema. This stage continues to grow and expand the colony. Four. Leafy stage development. The secondary protonema develops lateral buds. These buds act as the starting point for the next phase of development. 5. Mature leafy gametophyte. The lateral buds grow upward to form the mature leafy gametophyte, consisting of an upright slender axis bearing spirally arranged leaves. It attaches to the soil via multicellular branched rhizoids and will eventually bear the sex organs, Antheridia and Archegonia. Six. Release into water. Antherozoids, male gametes, are released into water, which acts as the essential medium for their transport. Seven. Fertilization. The antherozoids swim and come in contact with the Archegonium, the female sex organ. An antherozoid fuses with the egg to produce a diploid zygote. 8. Delayed meiosis. Zygotes do not undergo reduction division, meiosis, immediately after fertilization. 9. Sporophyte development. Instead, the zygote undergoes mitotic divisions to produce a multicellular body known as the sporophyte. 10. Dependency on gametophyte. The sporophyte is not free-living. It remains permanently attached to the photosynthetic gametophyte to derive its nourishment, water and nutrients. 11. Spore formation. Some specialized cells within the sporophyte, spore mother cells, undergo reduction division, meiosis, to produce haploid spores. 12. Germination. These haploid spores are released and germinate to produce a new haploid gametophyte, completing the cycle.